This is a test that we do routinely in our lab. Hi, this is Aatiya here. Welcome to my channel. Today I would be discussing one procedure called as total Kjeldahl nitrogen. We will see how to go about it. Let's get started. When any kind of treatment is done, whether it is wastewater treatment or any kind of treatment is done on food, it becomes important to note how much of organic nitrogen is present to see whether the treatment has worked or no. One of such nitrogen is Kjeldahl nitrogen, and we will see how to estimate this Kjeldahl nitrogen. Before that, what is Kjeldahl nitrogen? Kjeldahl nitrogen is the amount of organic nitrogen which is present in the waste or in the food or in anything. So today we will see how to quantify this organic nitrogen using a method called as Kjeldahl nitrogen method. There are three steps for this. Basically, digestion, distillation and titration. Technically speaking, organic nitrogen cannot be determined directly, and hence we require these three steps. Moving on to the first step, which is digestion. Digestion, as the name itself suggests, it basically involves breaking of nitrogen bonds from the sample. So what is done over here is the sample is digested along with sulfuric acid. We use fume hood chamber. It can be digested otherwise also. Basically, the only thing is that there should be an outlet for fumes, because this digestion procedure gives lot of strong fumes. A temperature of around 450 degrees Celsius is maintained. So we need high temperature to break all the nitrogen to nitrogen bonds. The catalyst over here usually which is used is potassium sulfate and copper sulfate. What it does is it helps in increasing the boiling point of sulfuric acid. When the boiling point of sulfuric acid is increased, it results in more amount of digestion. This whole mixture is cooled and is then converted and is then transferred to the distillation unit. Now here sometimes selenium or titanium, they are also can be used. Copper sulfate is slow to digest. But what happens is basically other mixtures containing titanium and selenium, they often result in generation of hazardous gases. So that's why we are using this mixture. So all the nitrogen that is broken down gets converted into ammonium sulfate. So the nitrogen gets converted into ammonium, which is then processed by distillation unit. So now we are done with this step. Okay. Moving on to distillation. As we know, distillation is the separation of components from a liquid. So if in liquid several components are there, we are trying to separate it. So what we have in the liquid which is supposed to be processed by distillation is ammonium ions. So we need to separate these ammonium ions and quantify how much of nitrogen is present. So that will happen in distillation process. Here an alkali is used. Generally the alkali is used is NaOH, sodium hydroxide. We use around 40% of sodium hydroxide over here. So what sodium hydroxide does is it helps in the conversion of ammonium ions to ammonia, which is then retained in a solution. That solution which is used is generally boric acid, but in its place hydrochloric acid could also be used. The purpose of using boric acid over here is it helps in capturing the ammonia which is generated in the solution. These are the two reactions associated with this particular step. First is addition of alkali. Second is how it is captured as ammonia. Now this is an equilibrium reaction. There is forward reaction as well as backward reaction in equilibrium. So basically by the end of this step we have ammonia into the solution of boric acid. Now the next job is to quantify how much of ammonia is present. That will directly give you how much of organic nitrogen which is present in the sample. And with that we are done with this step. Moving on to the next step, that is titration. So now our job is to quantify how much of ammonia which is present. In distillation step, if the receiving solution was boric acid, then the titration requires methylene blue and methyl red as indicator, a mixture of methylene blue and methyl red prepared in ethanol. In distillation step, the receiver solution was hydrochloric acid. Then the indicator which is required is methyl orange, and this is titrated against 0.01 normal sulfuric acid. How do you determine the color change? Before starting distillation, whatever color was there before starting the distillation, the same color needs to be observed after titration. Then you can say that the color change is observed, whatever it is. So let's move on to the calculations. How we are going to quantify how much of percentage nitrogen is there from the burette reading, which you get from your titration. So we are done with titration step. Now we are moving on to calculation part. So percentage nitrogen is calculated by 14 into normality of acid into burette reading into 100 divided by gram of sample into 1000. Now this 14 is a constant which is used over here for Kjeldahl nitrogen. Normality of acid, if you have used sulfuric acid 0.01, then that would be 0.01. Burette reading is the reading which you get during the titration. So usually we take 3 readings and then we take the average. So that average needs to be inputted over here. Gram of sample is how much of sample you have taken during the digestion process. For example, you have taken 2 grams or 1 gram of sample. Then in such case that you need to input over here. And this will give you how much of percentage nitrogen which is present. So from the sample we have deduced how much of percentage nitrogen is present in that sample. This parameter is required, especially if you are treating the sample with something like before treatment how much of percentage nitrogen is there, and after the treatment how much of percentage nitrogen is there, that you can deduce using this method. Thank you.